Hey, well, welcome everyone. My name is Warrick Inder. I'm an endocrinologist here at the PA Hospital and also have an appointment with the University of Queensland in the Office of Medical Education. But today I have the pleasure of doing a talk about research interest of mine, glucocorticoids. And I understand the idea of a seminar such as this is you get a clinician and you get a proper scientist and we get together and talk about things of common interest. So without any further ado, I will be able to advance the slides. So that smiley person in the corner there is Professor Vicki Clifton, and Vicki and I first came across one another in 1994 at the Endocrine Society of Australia annual scientific meeting. Now this was actually the first Aussie meeting attended by a kid from Christchurch, New Zealand. Well, when I say kid, I was actually 30 and I just had my second child who was about 2 weeks old. But anyway, now this woman from Newcastle wins the ESA Junior Investigator Award, and she gets up in her speech and says what a cool dude her supervisor is. So she was always going to go places, that woman. And in fact, in 2010, she became the first president of ESA in its 53-year history. And by the way, she appointed that former kid from Christchurch the treasurer. So we've known each other for a while. Now I came to Brisbane in 2015. At that stage, in 2011. At that stage, Vicki was still in Adelaide. But by 2015, we both ended up here in Brisbane. So what's the common ground? Well, we're both interested in glucocorticoids, and apologies to those who thought this was all going to be on asthma. Vicki does have an interest in asthma. Some of the participants in some of my studies have had asthma as well. But my main clinical expertise is pituitary and adrenal disease, and I've got a background of researching various perturbations in hypothalamic pituitary adrenal axis function across a wide range of conditions. The first paper I ever published was about athletes, and the second paper was about the effect of alcohol on cortisol. That was quite a fun study because we got everyone drunk and saw what happened to their cortisol. Now, Vicki is a basic science researcher with and she's going to talk about this obviously, but major interests in pregnancy, glucocorticoid receptor biology, and asthma. So the clinical issue that we were looking at and continue to do, is there a way that we could develop some measurable biomarkers of glucocorticoid activity that would be useful in a clinical setting? So what are glucocorticoids? Hopefully people will have heard of them. They're medications such as prednisolone, dexamethasone, hydrocortisone, and they're very widely prescribed in the population. So somewhere around 1 to 3% of the population at any one time will be on them. So if you've got 5 million Queenslanders, that's somewhere between 50 and 150,000 people knocking around Queensland on glucocorticoids. Now, at supraphysiological doses, and they're the kind of doses that most people get prescribed, there are multiple side effects. And here's my little Cushingoid person popping up in the side there. So all of these features can occur as a result of excess glucocorticoid. So there's a very characteristic clinical phenotype where they get a moon face, big rosy red face. They get abnormal fat distribution, particularly around the middle, so what they call truncal adiposity around your tummy, dorsocervical fat pad or buffalo hump, and also fat pads above the clavicles, which are called supraclavicular fat pads, funnily enough. You get thinning of the skin and muscle wasting, so there's sort of an increase in fat, reduction in muscle. You also get a reduction in bone, with predisposing to osteoporosis and fracture. Wound healing is slow, you get easy bruising, and probably something that's not that well appreciated is that with glucocorticoids, for pretty much any indication, there is a dose-dependent increase in mortality. So while it treats the underlying condition, it actually increases the risk of you dying than people not treated and that's even when controlled for severity of underlying disease. So supraphysiological doses of glucocorticoids in the long term kill people. Most dosing regimens are empirical, and there are a range of clinical manifestations. So not everyone gets this full-on, full-blown Cushingoid phenotype, and different people have different sensitivity, which is what we're getting towards. So what we really need is a biomarker of the actual glucocorticoid activity rather than just sort of pick a dose and hope it works. And that would be useful both for therapeutic drug monitoring, for people on steroids, and also for diagnostic purposes, looking at conditions of glucocorticoid excess, which is known as Cushing's syndrome, and adrenal insufficiency. And there is also an eponym for that, Addison's disease, when it's relating to the adrenal gland. So over about the last 5 years, the group I've been working with, we've published a few papers looking at potential biomarkers of glucocorticoid activity. This is the first one that was published in 2016, where we looked at a matricellular protein called thrombospondin 1. I dug up a very old paper from about 2000, I suppose it's not very old, but a 20-year-old paper from around 2000 that showed an in vitro study where thrombospondin 1 went up a lot in response to dexamethasone when PBMCs were bathed in that. So we actually showed that that was also responsive when humans were actually given glucocorticoids. I'm going to talk a bit more about the second study. This was published in 2019, where we actually looked at a ratio, so using the original thrombospondin 1 data, but also osteocalcin, which is down-regulated by glucocorticoids. So we were looking at a ratio between an up and a down-regulated protein. And then just published earlier this year, this was work done with Michelle Hill, who was formerly at the Diamantina Institute and now at the Berghofer. She had a proteomics facility, so what we were doing there was looking at discovery work, looking for glucocorticoid responsive proteins that weren't previously known. We managed to identify a number of proteins, but 3 that looked to be potentially clinically useful that increased in vivo when people were given dexamethasone, or decreased, changed in concentration. So what's this business about ratios? So a concept that a ratio between an upregulated protein or gene and a downregulated protein or gene would be more sensitive than either one alone. So if you've got a doubling of the upregulated one and a halving of the downregulated one, you've then got a 4-fold change in the ratio. So you're going to be able to pick up relatively smaller changes in glucocorticoid activity if in fact these markers reflect that. A common example in endocrinology of a ratio being in clinical use already is an aldosterone to renin ratio, which is commonly used to screen for primary aldosteronism in people with hypertension. And I think one of the other things to bear in mind is that ratios overcome the large or potentially large inter-individual differences at baseline. And that's particularly so with genes. And what we found, for example, with thrombospondin 1, was there was about 100-fold difference in normal volunteers between the baseline gene expression as determined by RT-PCR. So when you're expressing a ratio, one's going up, one's going down, it does sort of take that absolute concentration away. So these graphs indicate the data from the thrombospondin 1 to osteocalcin ratio paper. You can see in the left-hand side panel here, these were people who had received a dose of dexamethasone. The white circles are the baseline. They had the dexamethasone at midnight. They had blood sampling then 8, 12, and then 16 hours after the dexamethasone dose. And you see there that there was a significant increase across the board at all time points, but in particular the midday one was probably the most consistent of the 3. It's starting to wear off a little by 16 hours. We also looked at baseline levels in people who were completely normal, healthy volunteers versus people with Cushing's. It's a state of endogenous cortisol excess. And you can see there in the middle panel that, again, this is a log scale, so quite significant elevation in the thrombospondin 1 to osteocalcin ratio in Cushing's. The best, most discriminative time point was at 8 AM. And when you look here, we have a 95% sensitivity and 100% specificity for this particular distinction between normal people and people with Cushing's. That is as good as or better than any of our current diagnostic tests for Cushing's. So in summary then, when expressed as a ratio, rises significantly after a single dose of DEX, distinguishes Cushing's from normal. It's also, I haven't shown the data, but elevated in patients on moderate prednisolone doses. And it also goes up in people with adrenal insufficiency on so-called physiological replacement doses. So what about genes? So we've also identified 49 glucocorticoid responsive genes. And I see Adam Ewing was in the audience. He's done a lot of the analysis here. We're just getting that tidied up, ready to hopefully publish. So using a combination of gene ratios, we can actually distinguish normal individuals from those with too much or too little cortisol. There's a separate gene signature if you're taking exogenous prednisolone compared to having endogenous Cushing's. And also in the acute setting, it looks different for someone who's had a slug of dexamethasone compared to someone who's had synecthin. Now, synecthin is synthetic ACTH, which actually stimulates your endogenous cortisol from the adrenal. So while the net effect is an increase in glucocorticoid activity, the gene signature looks different across these various conditions. So what we're doing and what Vicki is doing with us is a new study to compare people weaning off prednisolone. We're doing a trial of the best way to wean prednisolone, healthy volunteers, more people with Cushing's and with adrenal insufficiency. And our hypotheses really are that the gene signature and these circulating proteins will provide an indication of total body glucocorticoid activity, which changes hopefully in a dose-dependent manner. And also looking at glucocorticoid receptor isoform expression, correlating that again with total body glucocorticoid activity and as a major determinant of activation and repression of these glucocorticoid responsive signaling pathways. Like most things, it's more fun when you do it with someone else. Research is better when you're working as a team. Complementary knowledge and skills. The project is going to provide lab and transitional experience to a recently minted, very fine fellow who's in the audience, Dr. Jack Lockett, who's doing a PhD with Vicki as his primary supervisor and me as the tagger. I want to pay some tribute to the TRI and to, in particular, the clinical research facility as well for providing such a state-of-the-art environment for us to work in. And also thanks to the Metro South Research Support Scheme and CERTA for funding of the project. And now it's like tag team wrestling. I'm going to pass you on to Vicki for phase 2.
Thank you, Warrick. So my area of interest that intersects with Warrick relates very much to the coexistence of stress and inflammation. And Zakar and I, Dr. Zakar Saif, who's in the audience, we have been working mostly around the placenta, pregnancy, and asthma. Glucocorticoids are well known for their ability to suppress inflammation and play a very central role in balancing immune homeostasis throughout the day, but also exogenous steroids, which Warrick has talked about, are used to inhibit inflammation in numerous diseases and infections, asthma being one of the ones that I'm particularly interested in, but also in relation to the treatment of inflammatory disorders, and also for the treatment of cytokine storms, which you actually have been looked at in quite, well, not as much detail as yet in the lung of COVID-infected patients. So we were interested in the combination of high circulating glucocorticoids and inflammation and what regulates that effect. And we know in many pathological conditions that there is high circulating or hypercortisolemia and inflammation in combination. On the right here, we're just looking at an example of that with post-traumatic stress disorder, where you have low-grade inflammation that persists, that influences brain function, while you also have hypercortisolemia, which also persists. In many syndromes, we call this glucocorticoid resistance, but the mechanisms that drive that resistance are not well understood. In pregnancy, we know that in the placenta, inflammation can persist even after several high doses of antenatal steroids such as betamethasone. So in this particular study, women were given repeated doses of betamethasone for fetal lung maturation as they were at risk of preterm delivery. And looking at the histopathology of the placenta after delivery indicated that markers of inflammation persist even though there were those sledgehammer doses of glucocorticoid being given to the mother. We also see it in animal models where you have inflammation induced into the rat placenta, which is exacerbated actually by the addition of glucocorticoids rather than being suppressed by the addition of glucocorticoids. In 2008, actually at an Endocrine Society of Australia meeting, John Sadlowski presented a very interesting talk on glucocorticoid receptor isoforms. And this actually set us on our current pathway in terms of examining the role of those isoforms in pregnancy and in the placenta. He reported that there are 5 splice variants: gamma, A, P, GR alpha and GR beta. And each of those splice variants have exon 2 retained in the gene sequence, which has 8 translational start sites. And those 8 translational start sites mean that each splice variant potentially has 8 translational isoforms. This makes a lot of sense because nearly every cell in your body expresses glucocorticoid receptors, but you don't want every cell to respond exactly the same way. And part of that is conferred by the presence of different glucocorticoid receptor isoforms. We were very interested in what was going on in the placenta and expecting just to find a couple of isoforms that may be expressed in the human placenta. But in fact, we found that there were 13 different isoforms, several of them that were known and characterised isoforms in other cellular systems, and some that were currently unknown but could be potentially translational isoforms. The glucocorticoid receptor isoforms varied in relation to fetal sex, maternal complication, gestational age, birth weight, and cell type. And we went on to find that these GR isoforms are conserved across species, and we've now found multiple GR isoforms in the mouse, sheep, guinea pig, and rat placenta. The function of these receptors is currently not well known, except for GR alpha A, which is the one that everybody looks at and measures. And its role is specifically around the inhibition of inflammation, growth, and nutrient transport. But the other ones, we really don't know what they're doing. We've been particularly interested in what regulates the growth of a fetus, especially in situations where the fetus isn't growing to its full genetic capacity. And we see that in babies that are small for gestational age. And these are babies that are less than 2,500 grams when they're born at term. And it's been previously reported that there is an increased level of inflammation in these placentas and increased concentrations of cortisol. When we looked at the glucocorticoid receptor isoforms, it was identified that GR alpha D1 was one isoform that was specifically upregulated in the presence of a small for gestational age baby. And this started to raise questions about whether this particular isoform may have a role in modulating that hypercortisolemia and inflammation. So Zaka started a series of in vitro studies where GR-alpha D1 and D2 were overexpressed separately in a trophoblast cell line. What we see here is that D1 and D2 are actually located in different parts of the cell, with D1 being closely located to the nucleus, while D2 is actually spread throughout the cytoplasm. With the overexpression of these isoforms, we then measured baseline mRNA concentrations of different genes that are associated with inflammation. And in the blue bars are the cells that were overexpressing D1, and the dark bars are those that were overexpressing D2. And you can see that baseline expression of inflammatory cytokines and other factors are significantly upregulated in the presence of D1 rather than in the presence of D2, and starts to point towards GR-alpha D1 having a role that may be associated with hypercortisolemia and inflammation. The work on this topic is ongoing in our laboratory, but Zaka said we should start to look at the immune system. And it was time to work probably beyond the placenta. So when we moved to Brisbane, I came to MARTA Research as an NHMRC Senior Research Fellow. And I was working with Joanna Barclay, and I met Adam Ewing, and I already knew that Warrick was here, so Warrick's WI there. And we started to have a few conversations about what I was doing and what Warrick was doing, and identified that there's probably a common interest in glucocorticoids, but probably a place that we can actually go to start to work together. And we started looking at glucocorticoid receptor isoforms in PBMCs in 2017. And it's taken us some time to get our act together, but finally we've recruited Jack, and we're now busily working on glucocorticoid receptor isoforms and their role in glucocorticoid sensitivity. So this is a little bit of work that Zaka's done and that Jack's been analysing. And what it's identified is that the PBMCs collected from patients with Cushing's, patients treated with prednisolone, patients with adrenal insufficiency do vary across pathology, as well as in the types of isoforms that are expressed. But you can see there are multiple isoforms that we find in the PBMCs. And Jack is looking at the relationship to glucocorticoid receptor isoform expression and downstream signalling pathways. And that's, it looks pretty bad on the screen here, but we're starting to look at that in collaboration with Adam and Warrick to examine whether there's a relationship between the glucocorticoid receptor isoform profile and downstream signalling pathways in PBMCs. So what do we gain from this collaboration? Well, Warrick wants a test that identifies glucocorticoid sensitivity, and we want to know how different glucocorticoid receptor isoforms work together to determine a specific cellular response to glucocorticoids. Warrick had the patient groups, biological samples, the gene expression data, and also the funding, which makes a big difference when it comes to doing a project. And we wanted to examine the glucocorticoid receptor isoforms and find ways to interpret glucocorticoid sensitivity in terms of the cellular receptor profile. And that's how we've come together and where we're heading. So I think it's a really nice collaboration. It gives a good example of how clinical problems can have a basic science component attached to them. It has led to funding support, it has led to student projects, it's enhanced our knowledge and understanding of each other's research focus, and it has the potential to translate into a clinical tool. But collaborations do require work because different disciplines often speak a different language, and it's important to connect in terms of knowing each other's language, understanding where each individual is coming from, and then actually finding a common pathway. You need to take time to talk and discuss potential projects and interests, and it's it takes time to find the funds to do those projects as well, but it's always worthwhile and fun. So thank you very much, and we can have time for questions if anyone has any. In the chat, Warrick, do you know if TSP1 and osteocalcin ratios reflects glucocorticoid activity in mice?
So no, I don't think that has actually been done. I mean, that was sort of a, for want of a better term, invention of our own group. So no one else, to my knowledge, has actually looked at it in any other species.
Thank you for that. I learned a bit. I'm not sure if Vicki alluded to it or whatever. What about post-translational modifications? Something like good old phosphorylation, which make a protein more active or less active or whatever.
Yeah.
And then I guess, looking for ratios in their status. Can you look into that?
Yeah, so we have been looking at the phosphorylation status of the glucocorticoid receptor isoforms, but of course there's many different post-translational modifications that can be made to the glucocorticoid receptor. The phosphorylation status is one modification that it determines whether the receptor moves into the nucleus or not. And so that's one of the areas that we have been focusing on. Was there a second part to that question?
No. I'm not sure. In the old days, you had to do radioactive assays. Do you have these days antibody specific?
Yeah, it's antibody specific. You can use Westerns to do it. Yeah. The thing about the glucocorticoid receptors is that all of the antibodies cross-react with all of the isoforms. So at this point in time, the most effective method is actually a Western blot, and there is no other way to distinguish the different isoforms of the receptor at this point in time. So that's a limitation.
Anything else in the chat?
No. All right.
All right, looks like we're off the hook. Thanks everyone for coming and zooming in.
Yeah, thank you. And we'll see you later. Okay.